This podcast is brought to you by Villanova University on iTunes U. Please visit us on itunes.villanova.edu.
Welcome back. First of all, I want to thank all of our panelists today for the outstanding talks on Mendel and his milieu, for the scientific legacy of Mendel, for the social, historical, and cultural implications of modern genetics. We're very disappointed that Dr. LaRusso and Dr. Green could not stay with us, but we have everyone else with us, so thank you so much for having made today such an amazing day. The panelists now are going to indulge us to focus specifically on the ethical implications of what we've discussed today. And they are joined by our Villanova bioethicists, whom I'd like to introduce to you today. And going towards my left, he will raise his hand, Dr. James McCartney. Dr. James McCartney is an Augustinian priest and an associate professor of philosophy at Villanova and an adjunct faculty at our law school. Dr. McCartney is a talented clinical medical ethicist. And most relevant for this panel, I call attention to his service on the professional staff of the National Commission for the Protection of Human Subjects Research in 1977 and 78. His new edited volume has just been released on the ethics of organ transplantation. But really, most importantly, all of Jim's accomplishments are surpassed by the fact that he chaired the search committee that hired me at Villanova. So thank you, Jim. I am going to pay that compliment forward, and I myself chaired the search committee which made the brilliant hire of Dr. Stephen Napier. Please raise your hand. Dr. Napier is the assistant professor of philosophy at Villanova and also an experienced and talented clinical bioethicist. He has been a certified institutional review board specialist and has written on bioethics and cognitive science in numerous academic journals and books. He is the author himself of a book on virtue epistemology. Together, we are the ethicists at Villanova who work in the philosophy department on issues in bioethics. It is my great delight now to introduce to you a bioethicist and accomplished scholar who is in fact a product of the ethics education at Villanova. Dr. McCartney and I take shameless pleasure in our modest role in the awakening of the interest in bioethics of Dr. Anna Smith Iltis. Dr. Iltis is a member of the class of 1996 College of Liberal Arts and Sciences. She was a presidential scholar at Villanova, an honors and philosophy major, and a bio minor. She holds a PhD from philosophy from Rice University, and she is professor in the Department of Social Science and Health Policy and director for the Center of Bioethics at Wake Forest University. She is widely published in this area of research ethics. And so when Father Ellis said we were going to do a symposium on Mendel, and we said we must focus, of course, on a session on ethics, we knew we had to invite Anna. She serves on several NIH data safety monitoring boards, and we are delighted that she agreed to join us today. So what's our plan for today? We want to do two things, really. We want to raise very briefly ethical considerations of selected issues in genomic medicine and research. And so to that end, each of the bioethicists will speak for 5 to 7 minutes only about a particular area in genetic research that raises ethical considerations. And then we will turn to a full roundtable discussion on the ethics of population-level genomic screening. And to orient both of these, we have provided you a case that we have designed. It's a fictionalized case, but it represents the kind of research that is being done and is actually based on an article that came out 2 months ago about a research study. And then what we'd like to do is open up that discussion of ethics of population genomic screening and any of the issues we brought up to you all to have a very inclusive roundtable. So you have the case. I'm not going to read the case, but I'll just highlight some certain areas from the case for you. This is a case to ascertain whether or not we could do accurate screening of certain monogenetic Mendelial disorders. Would screening of a population or the population actually be feasible? So it's a pilot study, and the method includes drawing blood on 1,000 healthy individuals and sequencing their whole genome, focusing only on 13 genes, that cause about 5 conditions. And then you heard about algorithms. The purpose of the study is to evaluate 5 different algorithms to see which one is best at identifying pathogenic or potentially pathogenic variants. And by best, what I'm talking about is the one that perhaps has a quote-unquote acceptable level of false positives and false negatives. Because if we're going to be screening people, we don't want it to be telling them they have something, they don't have something, and to be wrong in a significant portion of the time. So, as you know, in order to be in a research study, you have to give informed consent. So this informed consent asks people to give permission for their blood to be drawn and for their DNA to be sequenced. It gives the purpose of the study, discusses possible further use, and the fact of that the sample and the data will be de-identified, and the presence of minimal risk. It offers a modest stipend, and it talks about the conditions of withdrawal of consent for future research. And what's interesting is these conditions, we actually used the privacy and trust principles that are in the precision medicine initiative that is moving forward in our country today. In terms of data management, we said the samples are going to be stored by codes that's linked to the participant's identity. They'll be on password-protected computers with encryption, and only the investigators will have access to this identifying information. The sequencing data will be stored on the university servers. The samples and data may be available in the future for additional research, but not with identifying information. And if, by chance, a security breach occurs, investigators will make every effort to notify all affected participants immediately. So that is the crux of the case that we've designed for you. So now, my colleagues and I will raise for you 3 to 4 issues in ethics and genomic and genetic research, and then we will turn to a discussion specifically of the issue of the ethics of population genetic screening. I turn it now over to Father McCartney.
Just want to say a quick word about Anna Smith Iltis. She was a senior in high school when she took my Ethics for Healthcare Professionals course many, many years ago, and she was the brightest student in the class, and she came then the next year as a presidential scholar. I think I introduced her to Sarah Vaughan, who was director of the ethics program, if I'm not mistaken, at that time, and it went from there, and Anna went to the same university that Sarah Vaughan graduated from, so we've both had a great deal to do with Anna's development, and we're delighted to have her here.
About that.
I'm going to be very brief. I had an advisor one time that said, when you speak, stand up, speak up, and above all, know when to shut up. And the time is short, so this might not even take 5 minutes. But what I'm going to do is take the role of a person who's on a institutional review board reviewing this protocol, and especially for some of the informed consent requirements. Some of this is actually in the case itself. But I think it would be helpful just to see what someone on an institutional review board would raise. And then I think my colleagues are going to actually explicate this even further, but I'm mainly just going to hit the highlights. If I were a member of the Institutional Review Board, an IRB, reviewing the case study as a research protocol, I would require the following in order for the protocol to be in conformity with the standards for informed consent established by 45 CFR 46, sections 116 and 117, commonly called the Common Rule. First, it should be explained to subjects that their blood will be drawn, their DNA isolated and sequenced, and experiments will be conducted on 5 possible genetic tests to try to determine and/or improve the accuracy of these tests. A general explanation of the importance of accuracy and the reduction of false positives and false negatives will suffice, as long as the volunteer subjects understand that they are not undergoing the type of genetic testing that will provide them with accurate results. A statement that the purpose of the research has been explained and that the subjects have understood it shall be included in the consent form. Secondly, it should be explained to the subjects that the risks are minimal, a blood draw and very low probability that their confidentiality will be breached. Subjects will be given in writing the name and phone number of a contact person who can assist them in the unlikely event that there are negative effects, for example, infection or other types of negative effects. Researchers should be willing to pay the cost of any unlikely negative effects of the blood draw. How subjects' confidentiality will be protected should be explained in a way subjects can readily understand. If confidentiality should somehow be violated, subjects should be told that one of the researchers will contact them immediately to explain the nature and scope of the breach of confidentiality. The consent form should contain this assurance. Third, it should be explained to subjects that these results will yield some false positives and false negatives, but if the experimental results indicate that the subjects have a potentially pathogenic gene variant, they will be contacted and advised to meet with a genetic counselor and pursue genetic counseling for the gene or genes in question. Fourth, the subject should be told that their genetic samples may be used for future research, but they should be told, and the informed consent form include a stipulation in accordance with the privacy and trust principles of the Precision Medicine Initiative, that they may withdraw their consent for future research use and data sharing at any time and for any reason, with the understanding that consent for research use of data included in aggregate data sets or use in past studies and studies already begun cannot be withdrawn. The subject will be given the names of researchers or a website that can provide further information about the study at the request of the subjects. This is also the person or persons whom the subject should contact to withdraw their consent. I would also recommend the following. Because the provision of a $100 stipend might be coercive or at least skew the study towards persons from lower socioeconomic groups who might be more likely to participate in the study than others, I recommend that the stipend be lowered to $25 for participation in the study. Thank you.
Okay, thank you, Father Jim, for your comments. Thank you, Professor Brakeman, for inviting me, for hiring me. Okay. So I want to canvass again, my comments very brief. I want to focus on several challenges in meeting informed consent. So Father Jim outlined what you're looking for in informed consent. So I want to discuss briefly what makes getting informed consent particularly difficult for genetic research or genomic research. My cognitive labor here is made easier by previous work by Amy McGuire and Laura Biscow. They outline, basically you can see several ways in which, on the left-hand column, several ways in which genetics research is unique. I want to summarize here, there are basically 3 properties, okay? There's the identifiability of the information, okay? It's your DNA. Second property that's unique to genetic research is the durability. So the storage of your specimen or your information can endure for a length of time. And third, there's the involvement of other subjects insofar as genetic information implicates relatives. Okay, so these 3 categories, these 3 features of genetic research make consent to research and consent to genetic research particularly difficult. So taking the first one just to get descent to a few more details, the issue with indefinite storage and use of biospecimens, so what happens is the research never ends. Well, typically you think about consent in research as the subject is consenting to a particular research intervention with temporally indexed activities. The research begins and it ends, okay? Well, that's not the case, or not necessarily the case with genetics research insofar as your specimen or your information is stored indefinitely. Similar things can be said about collection of biospecimens for unspecified use. Again, you think of consent as consent to this intervention, but future use is unspecified. Logically speaking, you can't consent to a particular research project because it's unspecified. An inherent feature of genetic research is that the DNA is stable and it is unique. And here there are obvious issues concerning the breaches of confidentiality that were raised in the previous lecture. There's broad sharing of specimens. Again, the more the specimens and the data are shared and distributed, the risks of breaches of confidentiality go up, or at least make it uncertain. Limited control over downstream use. Again, this was brought up in the previous lecture. And here there are obvious concerns about this. Autonomy rights. I also put up here, and this is my addition to McGuire and Biscow's work, is that it can preclude a prevention of harm. So if the data is de-identified, and I now don't know if it can be, but if it can be, right, that would prevent any kind of return of results back to subjects if anything were to be found that is interesting or medically actionable, if that phrase can be made concrete. Limited to right to withdraw for the same reason, right? If it's de-identified, you kind of lose your control over your sample and your data because it can't be linked to you, again, if it can be in fact de-identified. And finally, the genetic information implicates family members, okay? And if something were to happen, this will be discussed. If information were to be found that implicates family members, you have, the issue here is that you have non-subjects, right? People who did not consent, yet their information is relevant, okay? So here are some models of consent to try to get around some of these unique features. Again, identifiability of data over a long range of time, its durability, and also its implication of family members. So the traditional human subjects model is you get specific consent for each research project, okay? Okay, I'll come back to that, maybe. Human subjects modified is that you would just get blanket consent from subjects and leave it at that. There's the donor model, and this, it's on the opt-out model. So you check the box on your application for the driver's license, right? You're opting in. This is a slight permutation where you have to explicitly opt out of having your blood or your samples used. But I want to focus on partnership model as it was canvassed briefly in the previous lecture again. There is First Genetic Trust is a company that has a web-based interface where subjects can track the research that's being done on their specimen. They can either extend or restrict their consent. I think that the partnership model improves upon the traditional human subjects model, right? It's not just getting consent for each use, but there's also a disclosure, right? There's a clinical use to implementing the partnership model, okay? It leaves it's a two-way relationship between the researchers and the subjects who give consent for their specimens. Okay.
I'd like to take up the third issue Dr. Napier raises, the implication to others of genetic research. An emerging controversy in genomic research surrounds the following. Do researchers have a responsibility to offer the patient's results to genomic relatives? And if so, under what conditions? And so in some ways, this is about privacy within the family. And does that even make sense? The question of whether relatives should be offered any of the participants' research results is challenging because we see on the one hand, first-degree relatives have 50% of their genetic material in common. So the nature of the data we get will implicate people other than the participant. On the other hand, ethical and legal approaches to informed consent, research protections, and privacy safeguards in the United States mainly focus on the rights and interests of the individual participant. So in the case we gave you, let's suppose John is a participant in our study, and he appears to have a pathogenic variant leading to a high likelihood of one of the conditions studied. He also has a twin sister. We may, of course, think that any obligation, ethical obligation, to disclose the results to the sister falls on him. Nevertheless, let's say in the course of discussion with the researcher, John states he isn't going to tell his sister about the results. Should the researcher seek her out and tell her? This is called active return of results. What if the sister on her own approaches the researcher to ask if John's results have implications for her own health or for her reproductive planning? Should the researcher respond to her? That's called passive return of results. Both of these scenarios are happening with frequency now. And while recommendations and policies are in place for handling this in the clinical arena, no guidelines for return of results to relatives for participants in genomic research has yet to be adopted. So what should we do? The ethical framework we have in place for research ethics includes a focus on three principles: respect for autonomy, beneficence or doing good, and justice. So in light of this framework, I say that there are three possible ways we could respond. The first would be mandatory return of results to relatives at risk. In this case, researchers would routinely return results to relatives actively. Under this view, the early detection advantages for potentially affected relatives supersedes the confidentiality and the choice of the individual participant. So this is going to put the principle of doing good and beneficence as a priority. There are, of course, at least two problems with this view philosophically and ethically. It undermines the nature of human subjects research, the protection of participants, and the respect owed to persons. And in the effort to benefit others, it really could lead to negative sequelae. In other words, in pursuit of doing good for people by providing this information, it could cause more significant psychological, social, or moral harms. And to use Dr. Allen's work, the harm to privacy and to the construction of self could be problematic as well. So let's look at the second one, return of results to relatives at risk only with the participant's consent. This view holds that while reasons to disclose to relatives are certainly understandable, researchers should not have this latitude. This view gives primacy to the respect owed to the autonomy of the person who is consenting to the research. They are the participant. There are at least two problems with this model. First, in research as in clinical practice, while confidentiality is a core value, it is not considered an absolute value. And there are times when patient confidentiality may be overridden or limited. Second, given that this data is relevant for the health of relatives as well as for the participant, if ever there were situations where confidentiality and privacy were more circumscribed, genomic research appears to be it. Where does that leave us? At a compromise, of course. The middle ground. Researchers should protect the privacy of the research participant and the privacy of results, thus leaning in favor of respecting autonomy. So researchers are not obligated, therefore, to return a participant's results to relatives. Yet saying that there's no obligation doesn't mean that researchers are prohibited from disclosing to relatives. In certain cases. Beneficent concerns could trump respect for autonomy. On this model, let's say, when a participant refused to disclose to relatives the presence of a highly pathogenic variant that the relative is likely to carry, for which there is treatment, and for which disclosure is likely to avert imminent harm, researchers may return results to relatives. How would this be determined, though? Frankly, that would be a judgment call. And as such, it seems like it would be really unfair to put this on any given researcher's shoulders. So a model of this kind would need the requirement, say, of an ethics consultation at the university or the institution to support such decisions. Wolf and colleagues, in fact, propose a model like this, and I think they have it mostly right. So what are the implications for informed consent? Clearly, at a minimum, the researchers in our case must have a policy about how they're going to handle disclosure to relatives prior to initiating the research, and ideally the informed consent should actually address disclosures to relatives in as detailed a fashion as possible to ascertain what kinds of information the participant wants shared and with whom. It should stipulate as well that a participant's data that meets these exceptional conditions may result in a disclosure by researchers even without their consent. Including this fact in the informed consent would allow people the most robust kind of consent possible. In other words, if they don't like the fact that that could possibly happen, then they won't be in the research. Finally, speculations about future frameworks. I end with some thoughts about the process. This third position is obviously not fully fleshed out and is an initial attempt to balance the protection of human health and welfare, respecting beneficence, with the fundamental duty to respect participants' rights to confidentiality and privacy, respect for autonomy. This model then addresses the challenge of disclosure to relatives using a framework that guides most of clinical research ethics. But to me, this field, genetic and genomic research, which is already, as we saw today, transforming medical care and research, is potentially, I'm making a big claim here because I only have 1 minute and why not, is potentially transformative also of the ethical frameworks that populate the terrain of clinical ethics and human subject research. These frameworks, like the one I just used, start from the idea of the radical, atomistic individuals whose rights must in fact be balanced with others like him and herself. Yet as genomic research shows, the individual as the primary moral agent or isolated self is not altogether descriptive of the reality of human existence on the physical level as well as on the social, psychological, or moral level. Will then ethical frameworks that place filial obligations, solidarity, and or responsibility to others as foundational to ethical evaluation come to be seen as more fitting of the lived reality of human existence? I would like to explore this idea further, but this, alas, is a different talk for another time. Thank you.
I'd like to thank Dr. Ellis and all the members of the committee that put together this symposium for inviting me, and of course my former teachers, without whom I would not have pursued my field of study and gone to graduate school and everything. And there are other people here who were also very important during my time at Villanova, so thank you. It's really wonderful to be back. I have to take a brief detour because my current institution, Wake Forest, received a mention a little while ago by Dr. Comfort. And yes, in fact, North Carolina's forced sterilization program was alive and well into the 1970s. That should scare you. It was not until 1977 that the law went off our books. And just in the past few years, the legislature has passed a law and funded the bill, which was important, to pay reparations to people who could be identified who had suffered under that program. If you're interested, there is a free documentary about that program available on our center's website. It's called Wicked Silence, and there will soon be a study guide there as well. But it's a documentary that talks to people who lived through that experience, and so it's a really great piece of work that was done by people in our documentary film program with support from our center. So now back to the topic at hand. Researchers have long faced questions about how to manage research data and biological samples that they collect. Today, however, the scale of genetic and genomic research raises new concerns and makes old questions and concerns more significant. As Dr. Osorio reviewed for us, the quantity, the volume, and the type of data that are being generated and stored is greater than ever before. And I'll note that as part of the Precision Medicine Initiative, there are discussions about cohorts, research cohorts of up to 1 million people in particular studies. And then when you talk about aggregating data from different studies, you're talking about vast amounts of data and stored samples. In addition to the new research scale, there's another source of complexity, and that is the ever-growing connection between research and clinical care. So if you think of Dr. Comfort's bridges, that adds another layer of complexity, and it's often very difficult to disentangle these issues. So these factors raise new and different questions about what should happen to research data and to stored samples in the future, and they add new dimensions to old questions. It's impossible to offer a comprehensive and detailed answer to the question, what should we do with stored data and stored samples, and how should we protect them? Because we have no idea what the future developments will be. So we can't offer a detailed plan, but it is possible and important, I would argue, to have a principled framework for developing ethical data and biobank management plans. This is the governance issue that Dr. Osorio mentioned. And to have a plan in place for revising those agreements as the need arises. So I want to begin by suggesting that answering the question, what ought investigators and institutions do with research data and stored samples, requires that we adopt a stewardship model. This is a word that Dr. Osorio used earlier. That was not planned, but thank you. And I want to suggest that that's really a sort of fundamental concept in how we move forward. Investigators and institutions, and that's important, ought to be good stewards of research resources. Why? Right? Well, simply, they're entrusted with those resources in terms of money, biological specimens, time, health information, trust, among other things. And in being entrusted with these resources, they are expected carefully and responsibly to manage them. One of the challenges in developing and applying a stewardship model is that investigators and institutions are stewards of both research participants' resources and society's resources. And sometimes, though not always, there can be tension between what it means to be a good steward of each of those. So stewardship with respect to participants' resources may involve a number of things, including, first, respecting participants' decisions and wishes over time. Many challenges here are related to the quality of informed consent and what it's possible to know in advance. These are some of the issues that Dr. Napier addressed. Respecting participants' decisions and wishes over time, however, also means honoring their right to withdraw consent from research in the future. And there are many questions about what exactly that should and can mean, but it has important implications for how data are managed and how samples are stored. I once, not too long ago, had an investigator, we were in a conversation about how certain kinds of samples would be stored and what kinds of rights to withdraw research participants should have. And an investigator just looked at several of us and said, if you think we're fishing through our freezers to get rid of someone's sample, I have news for you. So that's worrisome. When data, because we're promising people that they can stop being research participants if they want to. So when data and samples are stored over long periods of time, new scientific developments might change what it means to respect past decisions. For example, participants may be told that only results and incidental findings that are medically actionable, so for example, the 56 that Dr. Green mentioned earlier, will be shared with them. Results that are of unknown significance will not be provided. What obligations exist over time as new information emerges? For example, something that at one time was of unknown significance later is of known or suspected significance. For how long should participants be contacted? When data or samples are used by other investigators, does the same obligation to return results or report incidental findings apply? What data sharing agreements need to be in place to honor these kinds of commitments? As you heard from Dr. Green, a significant amount of work has been done on the question of returning results and incidental findings, but new questions routinely emerge. Second, respecting participants' privacy and confidentiality. While this is a basic requirement in human research and something we typically promise, as you heard, there are many challenges in this area, especially as we begin relating research data to other data. What happens in the face of the re-identification science and the reality of that possibility? As data storage practices change and re-identification science becomes a reality, it's possible that information that participants received at one time about how their information would be protected or how much risk of re-identification they faced changes. So what they agreed to no longer, the conditions under which they agreed to something no longer apply. How does that affect what investigators and institutions may do and what they must do? To be sure, some people have argued that we must stop worrying about privacy because privacy is dead, as was one of our commentators mentioned earlier. They maintain that there is no way to respect privacy, and we no longer live in an age when that's a reasonable expectation, and so we should simply forget it. And although people have written privacy's obituary, thus far participants typically are told, we will protect your privacy, so we need to know what that means. Third, respecting the time and effort of participants as well as the risks they assume. There are lots of things this could mean, but it includes protecting the integrity of the data and the integrity of the samples so that you don't waste your resources. Stewardship with respect to society's resources also can mean a number of things. Some overlap with the stewarding of participants' resources and some do not. First, investigators and institutions should maximize the utility of the data and the samples they have. There's some overlap here. For example, protect the quality and the integrity of your data and of your samples. But as Dr. Osorio mentioned today, there also are extensive expectations now with respect to sharing of data and in some cases of samples across institutions and in some cases with researchers in other countries. Extensive sharing of data may maximize the utility of that data and the extent to which good things can come from it, but it also increases the risk of breaches of privacy of participants, as well as the difficulty that participants face in trying to withdraw from research, or the possibility that they are participants in research that they find objectionable. In determining what information to share with other institutions and other investigators, there's a trade-off. The more information you give about research participants, perhaps the better the use of the data will be, but that also then raises the risks, increases the risk of breaching privacy issues. Second, some have suggested that the obligation to use society's resources well means that institutions and governments should not impose rules, regulations, or obligations that detract from the research effort or undermine research. For example, investigators should not be burdened with too many obligations to look for incidental findings, to return research results, and so on. So some people think that asking that of investigators detracts from the research effort, and that undermines the goal and the usefulness of research, and that that's something we should avoid. Participants and society entrust investigators and institutions with their resources. This gives them obligations to be good stewards. Interpreting those duties, however, is a much more complicated process. It involves a series of normative judgments about what ought to be done. Sometimes what it means to be a good steward of participants' resources will be in tension with what it means to be a good steward of society's resources. And resolving those tensions in itself involves making normative or moral judgments. Often tensions that are resolved among obligations are resolved by trying to balance obligations and goals. Ethical analysis involves deciding what factors are relevant. How do we decide when people say, well, we'll have to balance the tension? How do you decide how to balance the tension, right? Making those decisions about what do we consider, how do we consider it, and how do we assign significance to those different obligations all involves ethical judgments. They're the kinds of judgments that people often make without recognizing that they're making ethical judgments. The case we offered here, and much of the research that we heard about earlier today, generates significant amounts of data. Managing the samples and the data generated over time raises a variety of ethical questions. as we think about the vast amounts of data generated in genetic and genomic research thus far, and the efforts to connect these data with medical and other health data, as well as with data that are not traditionally health data, like your Google searches, to connect, ultimately the goal is often to connect genotypes and phenotypes, we see that this is a very small glimpse into the future. As our case indicates, we are now at a point at which we are having serious discussions about the possibility of pursuing population-level screening for Mendelian disorders. What I'd like to do now is turn to our panelists and invite you to consider some of the ethical questions associated with that aspect of the case. What are the ethical issues that we should be thinking about as we consider the possibility of population-level screening? If it's possible to pursue this kind of screening, ought we to do so? Why or why not? And how ought we to frame these discussions? So I will stop there and let you all participate in our conversation. So thank you.
Thank you, Dr. Iltis. And now we'd like to open it up for our panelists to give us some thoughts and for you all to participate with us about what do we think about if screening was feasible, ought we to do it, and what are some ways in which we should be framing this discussion?
Can I just say, I'm just an ordinary guy. I don't see why to solve one of the problems which has been brought out today, why don't you just simply raise the gift card to $200 and say you're never going to know anything? I mean, you have people on clinical trials for drugs and they don't know whether they're in the placebo group or the active group. A blind test. Same sort of thing. We're never going to tell you. We're going to do the work. You're never going to be informed if you've got a problem.
May I just say something quickly? That was the way much of genetic and genomic research initially was done. And there are a variety of reasons why some of the thinking has changed on that. I think one piece of it is researchers were finding that they couldn't sleep at night.
I think Dr. Green emphasized that in his talk today, that if you have information that could be life-saving or helpful to people, you have sort of an ethical obligation to communicate that. Now, this particular study doesn't claim that they have a potential genomic problem. It just says they ought to get further tested. So, but I still think that has to be done in terms of, being honest with people.
But you can remove that. You don't need to identify, the investigator doesn't need to know the identity of the person. So that problem is gone. The investigator, he or she, does not know that the one that is positive and has got a problem is that person, because the name is removed from the data set.
And that's certainly a way you can look at it, but another way to look at it is you do have knowledge that someone is at risk. And what are your obligations?
But we know.
Talking about the bridge between the clinical and the research.
But we know that someone's at a risk here. I'm at a risk. I know that. I've had to have a heart valve replaced. And people are at risk. We are all at medical risks.
But if we can help people, ought we to? That's the question of, should a researcher be engaging in that? Or is the researcher's primary duty to generalizable knowledge, protecting patients' rights, but they don't necessarily have that same duty to care that a clinician would? So this is that whole. Pilar, did you want to speak to that?
Oh, yeah, I was actually going to make exactly that point, is that I think it's really important that we keep that distinction, right? And that I have seen any number of cases in which research is done badly and cannot generate generalizable knowledge because the people involved are confused and they begin taking their clinical, they put on their clinical medical hat and elevate their obligations to patients above the researchers' imperative to generate generalizable knowledge. Now, I think one of the problems is we need to be really careful when we design our studies so that we don't put researchers in that conflict, because when we do, the sleep at night problem is often going to lead them to undermine the research. But honestly, when that happens, then we've potentially put people at various kinds of risk for no good reason, right? And so I think it's a real problem, but I also want to step back and say, we keep talking about, we know that they are at this risk. We're talking about low coverage sequencing here, using algorithms that are experimental. And I do think that one thing that happens with a lot of genome researchers is that we think we know a heck of a lot more than we do. And so I just want to sort of put that out as a caution, that feeling that I can't sleep at night does come from, I think I know that this person is at risk for some horrible disease. But honestly, I have to say, we've left it unclear as to what the 13 genes would be and the conditions would be. But people always talk about the BRCA mutations in the breast cancer genes. The population level risk for somebody who has had no familial increase of, breast, ovarian, prostate cancer in their family. Honestly, there's not one paper out there that has a good number. It's probably less than, well, there are some papers that would say in the 40% range, but I even think that potentially is an overestimate, right? And so, yes, if somebody has this family history of breast and ovarian cancer, and she's a woman and she has a BRCA mutation, then she could have an 80+% lifetime risk. But when we're doing screening of people who we don't know what their family history is, that's what screening is all about, then honestly, right now, we actually don't know how much risk they have, but I can guarantee you that a lot of geneticists feel like that's a life or death decision that people should, and I should return that result.
In support of what Pilar just said, I agree that the more global ethical problem with the research being proposed is that you'll end up having a lot of people now concerned about themselves in a way that they wouldn't have been otherwise. And I guess if because Jim imagined himself as being on the ethics committee which would decide about this, so if I just imagine myself as benevolent dictator, I think, well, what would I do with this kind of research? I'd press the pause button for a little while, and I would divert the funding in two different directions. I would divert some of it toward research into the kind of thing I asked Robert Greene about, where we would find out a bit more about the biochemistry and physiology, which accounts for why it is that in some individuals the mutation leads to the disease and in other individuals it doesn't. My suspicion is that it's not in the socioeconomic interests of people who are near enough to the tools to want to do that kind of research, and therefore the benevolent dictator needs to step in and make it done. The other direction which I send the research is towards education, so that when people eventually do learn that in their genome there is a bit of DNA which boosts their risk for X by 3 times, they actually have the critical resources not to have an exaggerated response. But that's difficult, and it's intellectually challenging to acquire that level of critical resource. So, as I say, I think, to me, what's problematic ethically about this kind of research isn't the issues about informed consent are a little bit downstream.
So if it's so difficult, as you say, to do, even sort of under ideal conditions, maybe the claim that you're making is a little stronger, that if it's so difficult, it should not actually be done because any amount of education might still not be able to overcome the fact that people are going to respond to risk in this way, and it doesn't really change their behavior, it doesn't really give them useful information.
I'm actually more optimistic about that than some people are. And because we're here to talk about Mendel and his legacies, one interesting question to raise in the light of the points that Robert Greene made in particular, where he said that the classical Mendelian disorders turn out to be thin on the ground. in 21st century biology. Now, one might ask, well, what might that mean for education in genetics? Why do we still typically start students off with Mendel and his P's, which, if one's learning about inheritance, the beauty of it for the teacher, but I think the problem for the rest of us, is that that gives students a core understanding about how inheritance works, which gives no attention whatsoever to developmental context and environment. And you might think, well, maybe it's not in our interest anymore to create students who, when they leave the classroom, can only remember blue eyes and brown eyes. We've actually started a teaching experiment at the University of Leeds where we've rewritten the curriculum, and we start not with Mendel and his P's, but with something like the contribution that genes make to the condition of a heart. Something genuinely complex and multi-causal, and then throughout the course, emphasize gene-environment interaction, not as a cognitive luxury item to be met later on and forgettable, but rather as the core message. And we've actually, not to go on about it at length, but we've assessed the attitudes toward genetic determinism of students at the beginning and end of our course as against students taking the traditional Mendelian course. And our samples are small, and so I won't hang too much on it, but our data suggests that whereas students coming out of a traditionally Mendelian course are just as determinist about genes at the end of it as they were at the start, students coming out of this alternative course are less so. And that seems to me the kind of result we should want in 21st century genomics and epigenetics.
What age are you talking about?
University.
University?
Undergrads. Yeah. Undergrads, yeah. Just a pilot study, and we hope to extend it. But it encouraged me to think that it is the case that you can, with the right kind of teaching, have students emerge who, when confronted with the latest Gene 4 report, are actually on their guard intellectually and want to ask questions about the genetic backgrounds and about the ranges of environment in which those claims for what the genes encode were established. So I'm actually more optimistic about that than some.
We'd like to open it up to anyone who'd like to contribute to or engage in this roundtable discussion.
Let me just raise a question. If indeed people are not educated to the complexity of expression of genes, should we have these kind of studies that people enter into it with the wrong impression that they're going to find out monogenic disorders that maybe won't be helpful in the long run because the expression of the gene might not be that powerful. So, I think that raises the whole issue of whether this study is even realistic or not. And actually, I'm moving toward your approach that if we're going to do this, we probably shouldn't tell them anything just because it's so uncertain.
So, but I think one of the things is that goes specifically to what genes and what conditions you actually pick. Because I think, when Robert gave that example of the ones that they put in the ACMG report, when he went back and looked at it, there was a reason why he said, look, over 80% of the people, in where we found a secondary finding had developed the disease. That's because they pick things where we already know that penetrance is very high, right? And I think if you're going to, at least we have a very good, we have a lot of evidence supporting that proposition. We still keep getting surprised, right? We still keep finding these situations where things have been reported in the literature as being clearly pathogenic, and we find apparently totally healthy people walking around with those variants. So, it's not perfect, but I think when, if we're going to think about any kind of screening, part of what we need to do is look at the evidence base for the things we would consider screening for. You don't just
Screen to screen.
Yeah, absolutely.
Yes, we have a question from the audience.
Thank you. Thank you for this wonderful presentation. I would love to hear your opinion with respect to how you preserve the element of voluntary participation and non-coercion, following on Dr. Comfort's lecture, when you have the informed consent document cover unspecified future uses for a specimen. And as you all think about developing an ethical framework here, shouldn't it be that you only are limited to use that specimen for the original purpose for which it was obtained, or for conditions that are reasonably related to that particular condition? So to me, for example, I worked in the state of Florida a number of years ago, where it was very clear that there was a specific law that said genetic testing results are deemed to be the personal property of the person who is tested. So I used to work at a hospital at the time, and we had to make sure that that was preserved by ensuring that that information did not make it into any shared network or drive or anything that would put it outside of the hands of the person's personal property with respect to those test results. So I'm wondering, as you're thinking about an ethical framework here, what can be done to ensure that you're not, you already have a lot of people distrusting the research enterprise per se, that you further exclude people from useful and helpful research by, because of the distrust of what these future uses may be. So if you could comment on that, I would really appreciate it.
Who would like to comment on that?
Okay. So I don't know if some of these ethical worries can be completely eliminated, but I did mention, and Dr. Osorio mentioned as well, more of a partnership model approach to consent. So this would involve recontacting the subjects for each research that's going to be done on their specimen or their data set. Now, the objection to the recontacting subjects is it's completely onerous to the researcher, et cetera, et cetera. But there are some attempts to streamline this process. For example, I mentioned First Genetic Trust, which has a web-based program where you can go on and you as the subject can find out what research is being planned or being done on your data set or specimen, and you can either restrict or extend your consent to those projects. And there's a web interface, and I don't know if there's any empirical studies that indicate that that's effective, if subjects really like it, but that seems to me, technologically speaking, one way to give more control and latitude to the subject, and at the same time reach the ends of research.
Yeah, so I think that's right. Holly Tabor and Mike Bamshat also have a completely nonprofit NIH-funded project where they're doing something similar. And so I do think that those kinds of models, particularly in communities where, if you're doing research in communities where there's a history of bad relations between researchers and participants, I also think that, for instance, right now, NIH is very much about pushing data sharing, but there are some times when it's just not worth it. So I work with a number of American Indian tribes, and there's a very significant level of distrust of federal government, of researchers. There are lots of layers of distrust. And I've been trying to get the institutes to make exceptions in various ways to their requirements that data would go into dbGaP or some data sharing repository, because you're absolutely right. And I think somebody else made this comment earlier that, we in our initial research project can make all kinds of promises and absolutely do phenomenal things to, for instance, maintain confidentiality and to do only what we said we would do in our consent form. Once it goes into a repository, in an interview that I was doing, one of the researchers said, well, it's basically in the wild. And I think that's true, and I do wish that, I think there are communities that have good reason for distrust, and our willingness as a research community to say sometimes doing some research in a more cabined way where we don't require the same kind of data sharing it's a long-term endeavor to build trust. So I wish there was more willingness to do that. Also, some of the technological approaches that I was talking about earlier, for instance, the distributed analysis. I know that some of the Strong Heart study, which is a heart disease study that involves a number of tribes, they've been talking with Lucilla's people at UC San Diego about whether they could develop the kind of computational capacity and database that would allow other people to send an algorithm in and do an analysis, but they wouldn't have to send their data out. And that way they would have, as a community, a lot more control over what was being done, and they would know what was being done.
Hi, I have a question. I want to go back to the issue about informing relatives of those who were screened. From my knowledge of genetic issues, what I see is that a lot of it is not only the genome, but a lot of social stress models that influence what genetic disorder you actually obtain. Say someone who has lung cancer, if their relatives didn't smoke, even though they may be predisposed for it, they would not most likely get that. And as far as we're looking now, if someone is screened and has a likelihood for a genetic disorder, they do not yet have that disorder, and then telling those relatives who also, we do not know their relative risk for that disorder either. Whereas taking models now with people who go in and are actually treated for lung cancer, for other genetic disorders, we don't have the discussion about telling the relatives that those people, that those patients have that disorder. And so the relatives, the relatives of that person would be even more likely, since that person already has that disease. It's not just they have a risk for that disease. So, and it seems like, at least in the patient contact-wise, that information is highly protected. So I feel like the genetic debate just kind of underlies that, and it's not really something completely new, like this very new information. These genetic disorders already underline what we're treating, and we already don't tell relatives of what we're treating, the patients that we're treating.
Thank you for that question. You're absolutely right that in research, if we find this pathogenic variant, that's a finding about the participant. It's not necessarily a finding about the relative, who would then have to go and get testing. So you're right, there is that that sort of disassociation, and that's why I said in exceptional circumstances. But in fact, in clinical context, we do have standards for guide that. So in 1998, the American Society of the Human Genetics actually said just this, that in fact, if you have a situation where someone has this condition, that there is an identifiable relative, that it is highly likely that this relative will also have this genetic predisposition, and it's something that you could treat. It's medically actionable, and that if you don't, it could actually undermine their health. So it would lead to morbidity and mortality. That researchers don't have, that physicians don't have a duty to warn, but they have a privilege to warn. And so that actually in clinical practice, there is that exception. Now, one of the things you raise is protected health data and HIPAA, and some research is covered by HIPAA, but some isn't. And what's also interesting is that if I have a doctor, and Anna's my sister, and she has a doctor, my doctor can talk to her doctor to get relevant information for my health care. That's allowed. So my doctor could find out this information, and derivatively, I would find it out too. So in clinical practice, and that's sort of, I think, what makes it interesting and thorny, in clinical practice, we actually do have some policies that date back before the human genome was sequenced to what do we do when we have genetic test results, should we tell relatives? And there is this general allowance that in these very exceptional circumstances that someone may do that. And so what I'm thinking is that maybe this would also apply to research as well, and what would be the circumstances in which it might be the case. And that's why I also struggle, though, as it sounds like you do too, with are the guidelines or the practice of research different such that because your main duty is the duty to generalizable knowledge while respecting patients' rights, there is a different set. So I see that tension. I'm not sort of saying, oh, 3, tie it up in a bow. I'm just bringing that up, but thank you for that question.
There's actually a more generalizable issue that you raise, even in the clinical context, not even with genetics. The Tarasov case in California Supreme Court said that a psychiatrist who felt that his patient was going to kill somebody and didn't tell her, and he did kill her, that there was an obligation to told her. I actually disagree with the case because it's a psychiatrist, but they came out with a very good principle. They said the protective privilege, which is confidentiality, ends where the public peril begins. So I use this in application where, let's say somebody has a sexually transmitted disease who's married, and he wants a physician to test the wife to see whether they have, and the physician he doesn't want the physician to tell the wife that he has a sexually transmitted disease because he didn't get it from her. Does the physician have an obligation to tell the wife? I believe he does, HIPAA notwithstanding, because that's somebody in direct public peril, and the protective privilege confidentiality ends when that person could possibly be harmed.
And the Tarasov is the duty to warn, whereas what we were talking about in clinical practice would be not a duty, but a privilege. We have time, I think, for one more question. Did you want to ask?
Thank you so much for your presentation. I really appreciated all of your comments. My question is directed to Dr. Osorio, though of course I welcome any other thoughts. I was particularly interested in your discussion of the health insurance, sorry, this is squeaky, the health insurance potential to do harm with protected health information. My immediate thought would be raising premiums for individuals who are diagnosed with potentially medical conditions or genetic diseases, but I was curious if you could offer thoughts on other potential implications of the violation of privacy with protected health data.
So I'm not sure I fully understood your question. If you're talking about the potential for like a disclosure of genomic or related research information to affect somebody's ability to get health insurance, is that what you're asking?
So my question was, I wasn't sure if that was what you were discussing, but you discussed potential misuse of information by health insurance. So it's like, where are you heading with this? What could go wrong, I guess?
Yeah, I don't actually remember saying anything particularly about insurance companies. Now, we have a statute, a federal law called the Genetic Information Non-Discrimination Act that is supposed to prevent health insurers and employers from engaging in various kinds of from doing things like conditioning your access on genetic information or an opportunity at employment or promotion on some genetic information. That statute only applies to health insurance and employment opportunities. It's enforced by the Office for Civil Rights in the United States, and they have brought enforcement actions mostly against not health insurers, but against employers who apparently have attempted to misuse genetic information. So that statute doesn't apply to things like long-term care and disability insurance. And I think it was actually another speaker who who sort of brought up insurance discrimination. Oh, and actually Professor Green, Robert Green, talked about people going out and buying long-term care insurance if they had an ApoE4 allele, and that being a concern of insurers, that when you find out that you have a higher than average genetic risk, then you run out and buy insurance. That's one of the reasons that the GINA statute doesn't cover long-term care and disability insurance. And so access to insurance still is a real concern for people, because even though various aspects of the law of health insurance have made that less of a problem in our country, there are other kinds of insurance that people care about a lot where those insurers might be able to use that information. So that's certainly one kind of potential use. Some would call it a misuse, some might not.
Thank you so much. This concludes our roundtable. I'd like to thank everyone. I'd like to invite Father Ellis up to say the last word.
We began planning for this today about last April, and finally to see it happen, and to happen so successfully, if I may say, on behalf of the committee that worked on this, I want to thank you all. And also, one of the things that we decided to do right from the beginning was to showcase our own faculty, and so we appreciate Father McCartney, Dr. Napier, and Anna, you're an honorary faculty member as well here at Villanova, so thank you as well. You know, one of the things that we always talk about here at Villanova is the idea of community as symbolized by the seal, which is a heart in the book that's very Augustinian, very appropriate since we're talking about Mendel to reference that. The book is about knowledge, it's probably scriptures, but we expand it to knowledge in general, and the heart, of course, about community and love. And so I was commenting on, during the break, I met up with Greg and Nathaniel, and we were with Simon, and we said, could we join you? He said, yeah, we went off, we looked at the Mendel statue and all that, but they were constantly talking about history of science the whole time. I said, you don't stop, you know, this. So the idea of community and the idea, the dialogue just continued on, and so it was great. And it's inspiring, really. So again, thank you, everyone, for your contribution to this day. It's been, I think, a great success, and I know it's been a long day, and you must be tired, but again, you have our appreciation. Thank you.